Right. So AP Bio. This is me testing out a new app. So let's hope it works. Called Screencastify. But anyway, what I'm going to do here is I'm going to go over just a quick review of Unit 1. So you might want to take notes while I do this. So if you want to pause the video, go get some lined paper and a pencil, and then come back and hit play. That would be a great idea. This is going to feel very fast because it's meant to be a super quick review. So don't feel bad if you feel super overwhelmed. It's meant to just be a really quick review so that then as we continue doing more in-depth review over the next few weeks, at least you've had a little bit of a reminder of what Unit 1 was about. And I'm going to be doing this for each unit over the next 2 weeks. So if you remember Unit 1, this was all the way back in August, we started by talking about the cell. So we talked about the comparing prokaryotic and eukaryotic cells. We talked about how the main difference is that prokaryotes lack anything that has a membrane. So prokaryotes do have a cell membrane, but other than that, they do not have any membrane-bound organelles. So they don't have a nucleus holding their DNA. But they do have ribosomes, they have DNA, they just don't have a nucleus surrounding it. Eukaryotes, on the other hand, have all sorts of organelles that they use to manage all of the functions inside. Then we went on to talking about the plasma membrane. And we talked about how the structure of that is special because it's made up of something called a phospholipid bilayer. And the phospholipid bilayer has hydrophobic heads and hydrophilic tails. Other way around. Hydrophobic tails and hydrophilic heads. Sorry, guys. It's my third time recording, so let's hope it works this time. So anyway, the hydrophobic tails, those because they're hydrophobic, meaning non-polar, they prevent anything that's polar from going through the membrane easily, like water. Does not have an easy time getting through the membrane. It needs a special channel. And so this is actually what keeps the outside of the cell separate from the inside of the cell, is this phospholipid bilayer. Then here, you might remember this image. We spent a lot of time talking about it in Unit 1. This was when we talked about hypotonic versus hypertonic, or about water potential. So what we see here is that this cell is an animal cell, and it's in a hypotonic solution, meaning a solution that has a higher water potential than inside the cell. And as a result, the cell is bursting because water is flooding into, the cell. Here down below, we also see a cell in a hypotonic solution, but it is a plant cell, so it is not bursting because the water is just filling up the cell. And then it hits the cell wall and it stays nice and firm. That's actually what keeps flowers upright. I'm looking at these flowers I have right next to me. Then we went on to talking about, or to applying this in the diffusion and osmosis lab. This was with pieces of potato and different colored solutions, and you practiced experiment design. So your independent variable, the thing that was different, was the molarity of the various solutions. This is what you were actually trying to figure out in the case of this lab. Your dependent variable was the mass of the potato. So you were looking at how the potatoes changed mass based on which solution they were in. Your experimental constants were many, but most of you had the same amount of solution in all of your beakers, or you had the potato in the solution for the exact same amount of time. And your control group, many of you had a control group that was distilled water. So you got some distilled water from a jug of distilled water I had. You added the potato core to that, and that told you, oh, hey, that is a comparison point, like a control group should be, because I know that this piece of potato should increase in mass the most, and I can compare that and see which of the solutions had a similar increase in mass, because that one is the distilled water as well. And the reason it should increase them in mass the most is because it has the highest water potential. distilled water has the maximum highest water potential ever because it has nothing else in it. And so that means that the water is going to flood into the potato, increasing the mass of the potato. Then we ended the unit by talking about cell reproduction, otherwise known as mitosis. We talked about how the cell cycle spends about 90% of its life doing interphase, and then that interphase is actually made up of 3 separate steps that all have checkpoints, and if something goes wrong with those checkpoints, that leads to cancer. And we went to that cancer research conference, if you remember. And then when we were talking about the steps of mitosis, we did our onion root tip lab, and that's where we first learned about chi-square, if you can believe it. It's that long ago. Okay. So that concludes that tiny lecture. I hope that was helpful. What is due on Wednesday at 11:59 p.m. is 2 things other than what you just watched. First, I want you to complete this Google form that I have linked right here. This Google form is called Something I Miss About You. I want to create a little bit of community, even though we're so far away from each other, because this is a particularly hard time in our school community. And so how this is going to work is you're going to write a note to someone else in our class. Let's say that I am Rashawn. I am writing to Mariana. So what I'm going to do is I'm going to write my name here. I write, my name is Rashawn. The person I am assigned to write to is Mariana. And something I miss about Mariana is, and then I would write 1 to 2 sentences about what I miss about Mariana. And then I'm going to post once, so this is due again on Wednesday, 4/8 at 11:59 p.m. because on Thursday morning, first thing when I wake up, I'm going to put all of these together and I'm going to share them with you on Google Classroom by 9 a.m. So you get to see all the nice little warm fuzzies. The other thing that is due at 11:59 p.m. is this Google Doc. So this is a case study on COVID-19 that gives you some real data from right now. And it is going to be pretty challenging because it's, like I said, real data. I'm going to let it load a second. But it also is really relevant because this is what's going on right now. And it's also great practice for the data analysis that's going to be on the AP exam. So I know it's going to be hard, but give it your best shot. And you're not going to be graded down if you get any parts of it wrong. Just give it a try. You're answering the questions right in here, but at one point it does tell you to use Desmos. So what you do is you click on the link I have here. That's going to take you to something called Desmos. You're going to put the numbers from the table into this table right here. You'll click here to say you want it to be dots. And then if I undo that I've done my graph, once you click that you want dots, then you're going to click here, and this is actually going to plot the points onto the graph. You don't have to take a screenshot or anything. You just need to have done the graph so that you can describe it in your Google Doc here. And then that is all. Don't forget to join the Zoom session on Thursday at 1:30 p.m. where I'm going to talk about updates to the AP exam. And here is a beautiful rose just to say I'm missing you. Bye, AP Bio. And this is where I try to figure out how to stop the video. Thanks. Bye.